Dear student, in this video, we are going to understand why chronic smokers are predisposed to emphysema. By definition, we can say that emphysema is characterized by irreversible enlargement of air spaces where distal to the terminal bronchiole, and it is accompanied by destruction of their walls without obvious fibrosis. But in recent studies, it has been shown that there will be small airways fibrosis in case of emphysema, and it will significantly contribute to airway obstruction. But this smaller airway fibrosis, it is totally different from chronic bronchitis. Okay, so emphysema, there will be irreversible enlargement of air spaces. Why? Because the elastic tissue of these areas, lung parenchyma, it will be lost. And where it will happen? Distal to the terminal bronchiole. From our knowledge of respiratory physiology, we know that the exchange zone of the lung, it mostly starts beyond the terminal bronchiole, and it includes the respiratory bronchiole, alveolar duct and alveolar sac. Here this is a place where actually the exchange of gases it takes place. Okay, so this space is also known as the acinus, and we can see in this picture, we can see this is a structure of a normal acinus containing the respiratory bronchiole, alveolar duct and alveolar sac. Okay, now according to which segment, according to the location of the segment where this loss of parenchyma is happening in case of emphysema, we can classify the emphysema into different forms. Most important form is the pan-acinar form. Here you can see the all part of this acini, that is starting from the respiratory bronchiole, alveolar duct, alveolar sac, everything will undergo the loss of elasticity and there will be enlargement. Okay, and this is known as pan. Pan is the entire, pan stands for entire acinus. Whereas another type is the centri-acinar. Here the central or proximal part of the acini comprising respiratory bronchiole, it is mostly affected. This centri-lobular type of or centri-acinar type of emphysema is commonly seen in case of heavy smokers, whereas this pan-acinar type of emphysema, it is commonly seen in case of alpha-1 antitrypsin deficiency. So in this figure, we can see that this is a normal alveoli and here this is a alveoli with emphysema. We can see the loss of the structure or the wall of the ear spaces, and this is the histological view where we can see definite enlargement of the ear spaces and the destruction of the parenchyma. Other than this pan-acinar and centri-acinar, we have two different types of emphysema too, that is the distal acinar and the irregular emphysema. Okay, but this pan-acinar and centri-acinar, these two are more important. Now we want to know, or we want to find out how this cigarette smoking is actually related to the emphysema. Now we all know that inhaled cigarette smoke and other noxious particles, they will cause lung damage. Most importantly, this damage is by the inflammatory reaction. Now this inflammation, it will result in parenchymal destruction and the emphysema. So the most important cause that the smoking it causes is the inflammatory changes at the parenchyma and the destruction of it. And the other causes may be the oxidative stress, and it will be increased due to this smoking, and that will cause increased apoptosis and senescence. Another is that there are certain genetic predisposition is also associated with emphysema. In persons with congenital alpha-1 antitrypsin deficiency, they are more prone to develop the emphysema, especially if the person is smoker. So the most important cause is the inflammation, secondly the oxidative stress, and thirdly, although there will be relative deficiency of alpha-1 antitrypsin, but those who are genetically mutated and have defective alpha-1 antitrypsin, so they are more prone to develop emphysema. These are the three pathophysiological causes. Come to the first cause, that is the inflammatory mediators and the leukocytes, how they are actually causing the lung damage. A wide variety of mediators have been shown to be increased in the affected parts. Okay, so smoking induced damaged part of the lung, there will be increased leukotriene B4, interleukin 8, tumor necrosing factor, and others. And these mediators are released by the resident epithelial cells and macrophages, and they attract more and more inflammatory cells from the circulations via some chemotactic factors. Okay, so initially there is damage and release of all these leukotrienes by the epithelial cells, and these chemicals will further attract so many other inflammatory cells, and they will amplify the inflammatory process by this pro-inflammatory cytokines, and it will induce the structural damage. Now we come to the second cause, that is the oxidative stress. How the cigarette smoking will cause the will increase the oxidative stress, and how it is causing the damage to the parenchyma or lung tissue, that we want to know. Now the substances like tobacco smoke, and we know that this smoke will cause some inflammation, and all of them will produce oxidants which may beget more tissue damage and inflammation. There are two opposing mechanisms that act inside our body. One will prevent the oxidative stress, another will enhance the oxidative stress. The NRF2 signaling pathway, it actually prevents or inhibits the oxidative states. Okay, so NRF2, it stands for the nuclear factor erythroid 2 related factor. NRF2 signaling pathway, we will discuss it in the next slide in more details. So if this signaling pathway is activated, that will prevent the oxidative stress induced damage. Whereas if the NF-kappa based signaling pathway is stimulated, that will help the oxidative stress to cause more damage. Okay, so if we want to prevent normally in our body the smoke induced oxidative stress, so this NRF2 signaling pathway has to be stimulated. Intracellular oxidants, they actually stimulate this NRF2 pathway. So there will be upregulation of this NRF2 pathway, and it will further upregulate the expression of multiple genes, and it actually encodes transcription factors that serves as sensor for oxidants. So there will be upregulation of multiple genes that will ultimately protect the cell from the oxidant damage. So oxidant is first released by inflammation, by smoke or anything else. So then that will activate or stimulate the NRF2. NRF2 will further upregulate, or it will act as a transcription factor, and it will upregulate multiple genes that will protect the cells from oxidant damage. Now this role of NRF2 gene, it is proved experimentally in mouse model 2. Here you can see that. Now a mice without NRF2, okay, if we see a mouse without NRF2, they are significantly more sensitive to the tobacco smoke than the normal mice. And NRF2 regulators and NRF2 target genes are all associated with smoking related lung diseases in humans. So mice without NRF2, they are significantly more sensitive to tobacco smoke than the normal mice, and these genetic variants, that is all these NRF2 regulators and NRF2 target genes, they all are associated with smoking related lung diseases in humans. This is the molecular mechanism how NRF2 acts. NRF2 translocation to the nucleus after dissociation from its inhibitory protein KEAP1, which is the Kelch-like ECH associated protein 1. Okay, so normally this NRF2 remains bound with this KEAP1, and when there is any kind of oxidative stress or like this, so there will be detachment of this NRF2 with this KEAP1, and this NRF2 then it will enter into the cell and it will bind to the specific region on the DNA, that region is ARE. ARE actually stands for antioxidant response element. Okay, so it will bind and activate this ARE in the DNA, and it will regulate the expression of several genes, including those involved in the oxidative stress responses. And we know that all these multiple genes will protect cells from oxidant damage. Now we came to know that what smoking does. Okay, it will deliver some oxidants, and normally in our body we have this NRF2 that will prevent this oxidant mediated damage. Okay, so how can emphysema happen in smoker? So in smokers with emphysema, we'll see that nuclear localization of NRF2 is decreased in macrophages, as well as the antioxidant response is also decreased. So when there is smoking, the oxidant is released. Naturally, the NRF2, it has to be stimulated and it has to protect. But what happens? There will be the nuclear localization of NRF2 is decreased in macrophages, as well as the antioxidant response will also be decreased. This altered antioxidant response is inversely correlated with airflow obstruction. Okay, so the first point, smoking will cause the inflammation, and it will release some chemicals that will further attract more chemicals and more cells, so there will be damage. Second, they will generate some oxidants. Normally this oxidant has to be neutralized by this NRF2, but the activity of this NRF2, it will be decreased. So antioxidant response is also diminished in case of smokers. That will also cause or enhance the further damage of the lung tissue. Now the third factor is the smoking will cause an imbalance between the protease and antiprotease. Okay, so protease, the example of protease is elastase. So the function of this protease or elastase is to destroy the elastic tissue. Okay, so normally in our body we have the antiproteases that will prevent the function of the protease. Okay, so there is a balance in between these two. But in case of smokers, this balance will be lost. So this is the picture of the perfect balance. So we know that these are the proteases or elastases released by the neutrophils, mast cells and macrophages. And there are several other factors which will combat these elastases. They have the antiprotease activity. For example, we have the alpha-1 macroglobulin. We have some secretory leukoproteases. Okay. And most importantly, the alpha-1 antitrypsin. This alpha-1 antitrypsin actually it leads to decrease the elastase induced damage. So elastase enzyme induced damage to the cell, it will be prevented by alpha-1 antitrypsin. Okay, so there will be a relative deficiency of this protective antiprotease, but in some cases there will be some kind of genetic basis. Alpha-1 antitrypsin, which is a protein, 90% of it is synthesized by the liver. It is secreted into the circulation and it protects the elastin in the lungs, that is destroyed by the elastage, and the normal level is, that is 104 to 276 milligram per deciliter. Okay, so this is the normal level of alpha-1 antitrypsin. Now, this alpha-1 antitrypsin is encoded by the proteinase inhibitor PI locus, which is present on the chromosome number 14. This PI locus is polymorphic, and in approximately 0.012% of the United States population, you'll see a homozygous for the Z allele. This is a specific genotype that is associated with markedly decreased serum level of alpha-1 antitrypsin. Okay, so some genetically predisposed person who are these Z homozygous, that is ZZ, their combination is ZZ, so they will have a decreased level of alpha-1 antitrypsin. Okay, so smoking is there, so oxidant is there, inflammation is there, elastage is there, all are trying to destroy the lung tissue, and there are several factors that will try to prevent them. Okay, one is NRF2, another this elastase mediated damage will be prevented by alpha-1 antitrypsins. Now, if these genetically predisposed persons who are having this homozygous Z allele, they are smokers. So 80% of these individuals will develop symptomatic pan-acinar emphysema, and which occurs even at an early age, and it is of greater connective tissue septa and at the margin of the lobule. So severe form of disease at the early age, it will occur in persons who are genetically predisposed, who are having ZZ, and they are smokers. Here we can see the phenotype. If it is so there is no increase or change in alpha-1 antitrypsin concentration, so no increase in emphysema. If it is MZ, it is not increase, so no emphysema. SS, no emphysema. Okay, so alpha-1 antitrypsin level is normal. If it is SZ, then mild increased risk of emphysema. If it is ZZ and null, then there is very high risk of development of emphysema. So this is the homozygous Z allele. Genetically predisposed persons, if they are smoker, then there is high chance of developing, 80% of them will develop pan-acinar emphysema. The symptoms will appear more early age, and the damage will be much more severe. So to summarize, the smoking or any air pollutants, that they will cause the emphysematous damage by 3 pathophysiological pathway. Most importantly is the inflammatory pathway. Second is the oxidative stress, and there will be relative deficiency of NRF2, that is that prevents the oxidant induced damage. And the third is there will be imbalance between the protease and antiprotease, and genetically predisposed person who have the congenital alpha-1 antitrypsin deficiency, they are more predisposed to develop the emphysema. So emphysema means destruction of the elastic tissue of the lung, the enlargement of the sac. It may be pan-acinar, especially in the exchange zone it will be affected, and the causes are, in case of smoking, will cause the inflammation, will cause the oxidative stress, and it will cause an imbalance between the protease and antiprotease. Thank you. So this is a reference, Romin's Pathology, 9th edition, page number 695.